Hello, and thank you for joining today. My name is Natalie Orr, and I am a product development scientist at Precision Nanosystems. Today, I'm going to present to you a novel lipid nanoparticle composition that is optimized for RNA delivery in vaccine applications. In case you're unfamiliar with us, Precision Nanosystems are a part of the Danaher Life Sciences family, and our company mission is to accelerate the creation of transformative medicines that significantly impact human well-being. We are headquartered in Vancouver, and you can see our headquarters here at the top with our new home starting 2024 at the bottom. And we are an end-to-end nanoparticle solutions provider with a focus on genomic medicines, as we firmly believe that they are the future. And we hold this belief as genomic medicines treat disease at their fundamental level. Depending on the specific gene target and mutation, genomic medicines can be used to silence genes, express proteins of interest, either to replace them if inadequately produced by a patient, or to elicit a specific immune response, such as seen in vaccines. Genes may also be edited using CRISPR and other technologies as well. And these approaches allow for targeting any gene anyway. Importantly, genomic medicines are designed and not discovered. They're validated, they're manufacturable, and thus are ready for mainstream use. So despite these clear advantages to genomic medicines, there are some challenges associated. Firstly, nucleic acids do not readily enter cells and are highly unstable, and so require a delivery vehicle to ensure that they get to the cells where they are needed to exert their effect. Secondly, genomic medicines are complex drug products and are traditionally challenging to develop and scale up. To meet these first two challenges, a highly skilled and diverse team are required with a wide range of skills targeted to each specific area of the development process. Finally, with each different application, there are different needs, ranging from highly specific personalized medicines to applications for mass administration, each with their own specific set of challenges. So despite these challenges, rapid and successful genomic medicine development is possible and has been showcased by the COVID-19 vaccines, which really were the game changer in the pandemic, with over 13 billion doses given worldwide. The timeline of the Moderna vaccine development is shown here, with the first trial participant vaccinated only 66 days after the SARS-CoV-2 genome was posted online. And although rapid, this response was made possible by decades of knowledge and experience built prior to the pandemic. So what is needed for the successful development of genomic medicines? The requirements can be summarized by 4 pillars. The first pillar is a requirement to define a specific gene target to silence, express, or edit, followed by the design of the active pharmaceutical ingredient, which in genomic medicines will be a nucleic acid. This is followed by designing a delivery carrier or vector, such as a lipid nanoparticle or LNP, to protect and transport the payload into cells. And finally, a scalable manufacturing solution is needed. Our product offerings, in addition to the expertise and experience that the Precision Nanosystems team has to offer, can help address these challenges that are associated in the development of a successful genomic medicine. So Precision Nanosystems, we offer proprietary manufacturing systems known as the NanoAssembler platform. And these instruments can reproducibly formulate genomic medicine drug products at both small and large scales, as we will discuss later in the presentation. At all scales, the next-gen mixer is used to mix 2 streams of liquid, typically of different polarities, in a time-invariant and rapid manner, resulting in the formation of the drug product. To complement the NanoAssembler systems, Precision Nanosystem also offer research use only kits, known as the GenVoy family, that are used to enable and accelerate the development of genomic medicines by lowering the barrier to entry and making everyone a genomic medicine developer. The GenVoy product family have been validated in various different applications, including vaccines, gene therapy, and ex vivo cell therapy in human T cells. But is there still a need for LNP-based genomic medicines now that the COVID-19 global health emergency is over? And yes, there absolutely is. A report published by Nature Reviews looked at both the current and future landscapes of LNP-based medicines, looking at clinical trials that are investigating these medicines as of December 2021. Here in the graph on the left, we can see that the majority of these are in vaccine applications, with gene replacement, control, and editing also being investigated, but to a lesser extent. Looking at the market forecasting on the right-hand side, we can see that over the next 10 to 15 years, LNPs will remain a core tool in the in vivo delivery of genomic medicines, especially in vaccine applications, with other applications also gaining traction. So with this in mind, we're developing a new offering to add to the Genvoy product family using Precision Nanosystems' own ionizable lipids that are available for clinical evaluation. So the aim of this presentation is to share the development process of a new LNP lipid mix composition optimized for in vivo delivery of RNA. To achieve this aim, we screened and selected the ionizable lipid and LNP composition using the physicochemical characteristics, the in vitro potency, and in vivo immunogenicity to guide our choice. We then optimized the formulation process parameters of the lead candidate, determined the resulting LNP storage stability, and investigated the scale-up potential. So in this first section, I will describe the selection of our lead lipid mix composition. All LNPs were formulated using a nanoassembler Ignite, pictured here in the schematic diagram, with a next-gen mixer, followed by buffer exchange, reconcentration, and sterile filtration. Process parameters are shown here in the table and were optimized for the lead LNP composition, although for brevity, this data has not been included. To highlight some important formulation parameters, a flow rate ratio of 3 and an entropy ratio of 8 were used throughout. The first step in the process was to select the ionizable lipid to be used in the LNPs, which is of high importance as it is the main component that affects both the LNP formulation and structure, as well as the transfection of cells and resulting biological activity. At Precision Nanosystems, we have a diverse library of many novel ionizable lipids. Based on the ionizable lipid structure and characteristics, alongside extensive screening, we have identified key lipids for different applications. After selecting the ionizable lipids based on the desired application, they can then be used in combination with other lipids to prepare LNPs. Using the screening process of the lipid library that I just described, the in vivo application hits were selected to formulate LNPs with a self-amplifying RNA encoding for the SARS-CoV-2 pre-fusion spike protein. Using these LNPs, we immunized biopsy mice with 1 microgram of RNA intramuscularly, followed by a booster dose on day 28, and determined the antigen-specific IgG levels in serum using ELISA. Looking at the graph on the right-hand side, you can see that all LNPs showed a strong immune response on day 42, highlighting the utility of the ionizable lipid library and screening process for different applications. You can also see that the immune responses shown here are comparable to those generated by LNPs prepared using the clinically relevant ionizable lipids SM102 and ALC0315 used in the Moderna and Pfizer COVID vaccines, respectively. Using these results, we were able to select the lead ionizable lipid for further LNP composition investigation. So to test different LNP compositions with the optimized ionizable lipid, we used the SARS-CoV-2 vaccine application with the same self-amplifying RNA and immunization schedule as the ionizable lipid screening process. Additionally, serum was sampled 6 hours after the primary injection to test for early cytokine release, and 2 weeks after the booster dose, spleens were harvested and the splenocytes were isolated. They were then re-stimulated with antigen peptides and the cytokine production was measured. So before jumping into in vivo testing, we wanted to determine the critical quality attributes and in vitro potency of the lead LNP compositions prepared with the lead ionizable lipid. The lipid nanoparticle composition options are endless, as many different lipids can be selected in various different ratios. Extensive screening was performed, and the options were narrowed down to 3 lead compositions. On this slide, we show that all 3 met the typical critical quality attributes of LNPs, with sizes of around 80 nanometers observed, a PDI of less than 0.2, and an encapsulation efficiency of greater than 95%. We then investigated the in vitro expression using HEC293 cells and Western blotting. Cells were treated with LNPs for 24 hours, and after uptake by cells and endosomal release of the RNA into the cytosol, the spike protein was translated and expressed. Cells were lysed, and then the protein separated by electrophoresis. The full-length spike and S1 subunit proteins were then detected using antibodies and chemiluminescence, and are shown in the plot here on the left-hand side. The spike protein band intensities were then normalized using the band intensities of the beta-actin loading control protein bands, seen in the graph on the right. From this, we found LNP1 and LNP3 to have similar potencies in HEC293 cells, while LNP2 expression was markedly lower. Moving on to the in vivo study described previously, we used the serum taken from the mice on day 21 and day 42 of the immunization schedule to determine levels of spike protein-specific IgG. While high median titers are observed for all LNPs, we did see larger spread in data for LNP1 on day 42. And somewhat unsurprisingly, we did not see correlation between the in vitro potency and in vivo IgG levels, with the humoral response to LNP2 being comparable to LNPs 1 and 3, despite showing a lower in vitro potency. At each time point, there was no significant difference in the IgG titers after treatment with the LNPs. We also investigated both the innate and adaptive immune responses. Differences in the early cytokine release profile are shown in the heat map on the left. Serum taking 6 hours after the primary injection was tested by flow cytometry to determine the concentration of specific cytokines. And while there was a strong response by CXCL10 at this time point, the cytokine profile in response to each LNP was comparable, if marginally higher for LNP1. The cellular immune response was determined by harvesting spleens at the end of the study, isolating the splenocytes and re-stimulating them with antigen peptide. The concentration of specific cytokines was then measured, and the graphs in the middle show a high interferon gamma response and lower levels of IL-4, comparable to those seen with the control group of mice, indicating a Th1 skewed cellular immune response. The heat map on the right shows the cytokine profile in response to each LNP treatment, and it shows that this was comparable between LNPs, and that the interferon gamma response was strongest out of all the cytokines investigated. Next, we tested the same LNPs in an influenza vaccine model. Using the same experimental design as was used in the COVID-19 application, we encapsulated mRNA encoding for hemagglutinin, a surface protein found on the influenza virus. We used a hemagglutinin specific to the H1N1 virus, specifically A California 07-2009 strain, and used both an unmodified and base-modified RNA for protein expression. While all other immunization schedule details were consistent with the SARS-CoV-2 study, the intramuscular dose of mRNA was increased to 2 microgram. So looking first at the physicochemical characteristics for the 3 LNP compositions prepared using both types of hemagglutinin mRNA, we find that all LNPs showed excellent size, PDI, and RNA encapsulation, meeting the typical critical quality attributes for vaccine applications. Next, we looked at the humoral response to LNPs after injection into mice using the immunization schedule previously discussed. We tested serum samples by ELISA to quantify the hemagglutinin-specific IgG titers, and we also tested the same samples using a hemagglutinin inhibition assay, which indirectly measures IgG titers by determining the highest serum dilution at which, when added to red blood cells in the presence of influenza virus, no clumping occurs or hemagglutinin is inhibited. In alignment with the SARS-CoV-2 vaccine application study, we find that LNP1 exhibited a large spread in humoral response for both mRNA investigated, although median IgG titers were comparable between all treatment groups. So looking at both the innate and adaptive immune responses after influenza vaccination, the heat map on the left shows a strong response by CXCL10, and in this instance, CXCL9. Looking at differences between treatment groups, higher cytokine responses were observed in reaction to LNPs with unmodified mRNA compared to LNPs with base-modified mRNA. And comparing the responses to different LNP compositions, no stark differences were observed. Similarly, the cytokine responses by restimulated splenocytes were comparable between LNP compositions with high levels of interferon gamma and low levels of interleukin-4 observed, indicating a Th1 skewed cellular immune response. However, splenocyte cytokine responses in general tended to be higher in LNPs prepared with modified mRNA compared to those prepared with unmodified mRNA, as shown in both the graphs and the heat map on the right-hand side of the screen. So to summarize this section, using the data generated in both the SARS-CoV-2 and influenza vaccine applications, we were able to select the lead ionizable lipid candidate and LNP composition for RNA delivery. The lead LNP composition showed excellent critical quality attributes with a variety of payloads, which differed by RNA type, chain length, and modifications. It also showed a high in vitro potency and a strong humoral and cellular immune response, which will allow the LNP to be used to accurately assess payload efficacy in the screening and optimization process. So after the lipid mix used in this kit was developed and optimized, we wanted to determine the LNP stability using a pre-developed cryopreservation buffer. So first, we'll discuss some of the challenges affecting LNP stability. So in general, very low temperatures are required to store lipid nanoparticles for a prolonged period of time, yet the optimal storage conditions, the storage buffer type, and the cryoprotectant appear to differ between formulations. The stability challenges of RNA-LNPs can be attributed to both the lipid nanoparticle itself as well as the RNA. Lipid nanoparticles are prone to both physical degradation, including aggregation, fusion, and leakage of payload, as well as chemical degradation of the lipids by hydrolysis and oxidation, to name a few. RNA, although protected from enzymatic degradation by the LNP, also faces both chemical and physical stability challenges. Chemical modifications of the bonds in the RNA molecule or oxidation of the bases may stop translation and can have huge implications on the efficacy of the RNA. Longer RNA tends to be more structured than shorter strands, and physical degradation or loss of these secondary and tertiary structures is also thought to affect RNA stability, as it can result in the aggregation of RNA molecules. In addition, the nucleotides used and any modifications can affect stability, as well as the lipid nanoparticle components, as these can form adducts with RNA and have been shown to affect stability also. So to investigate the stability of the LNPs developed in this study, the lead candidate lipid mix was used to formulate LNPs encapsulating both EGFP mRNA and EGFP self-amplifying RNA. In this study, we chose EGFP as it is a common green fluorescent reporter protein that can easily be detected. Additionally, we investigated both a conventional mRNA and a self-amplifying mRNA due to the differences in nucleotide chain length, with EGFP mRNA being around 1,000 nucleotides in length and EGFP self-amplifying RNA being around 8,500 nucleotides. The differences in chain length can be attributed to the extra sequences, here shown in the diagram in orange, that code for non-structural viral proteins that allow self-replication of the mRNA and amplification of the protein expressed. All formulation and process parameters used were consistent with the previous dataset. A pre-optimized cryopreservation buffer was used to store the LNPs for either 1 week under refrigerated conditions or for up to 6 weeks at -80 degrees Celsius. So looking first at the physicochemical characterization over the time period investigated, results for mRNA-LNPs are grouped together on the left of each graph and self-amplifying LNPs on the right. Each bar represents a different time point, as noted in the legend, and at each time point, LNP aliquots were removed from storage and the physicochemical characteristics were tested. And as you can see in these graphs, both mRNA and self-amplifying RNA-LNPs showed excellent stability over the 6 weeks time period, with no changes or trends observed in size, PDI, or encapsulation. We also investigated the in vitro potency of LNPs on HEC293 cells using flow cytometry. The graph on the left shows the potency of mRNA LNPs over time, and the graph on the right-hand side shows the results for self-amplifying RNA LNPs. The purple bars show the transfection efficiency, or the percentage of live cells that are expressing GFP, and the scatter plot shows the median fluorescence intensity, or the amount of expression from all GFP-positive cells. And what these graphs show is that the transfection efficiency and protein expression were maintained across all storage durations for both mRNA and self-amplifying RNA LNPs. Additionally, high transfection efficiencies of above 80% for mRNA LNPs and greater than 60% for self-amplifying RNA LNPs were observed. And with this dataset, we can confidently recommend that LNPs can be prepared with RNA of different nucleotide lengths using the materials provided with our LNP kit and stored for up to 1 week in the fridge and up to 6 weeks at -80 degrees Celsius before use, with no detrimental effects on the critical quality attributes. So moving on to the final section, we wanted to investigate the scale-up potential of the lead lipid mix composition. So despite the major advances in lipid nanoparticle technology and the large-scale commercial batch productions that made the global COVID vaccination campaign possible, challenges in the scale-up of LNP vaccines remain. To successfully scale from the bench to clinic, LNPs prepared at commercial scale must maintain the critical quality attributes seen during bench scale R&D. Changes in both the upstream and downstream processes, known as critical process parameters, during formulation can have huge impacts on LNP characteristics and the biological activity. And changes in these processes may be the roadblock on the path to commercialization. There are also added challenges with respect to the regulatory requirements with which the instruments must comply when commercial scale batches are prepared. For example, as large volumes of 100% ethanol are required to formulate, which is flammable at ambient temperatures, equipment must be certified to use in hazardous locations. In addition, software associated with the equipment should allow for electronic record keeping in compliance with FDA regulations. The equipment should allow for bioburden control, and it must also minimize extractable and leachable components passing into the formulation. To address the challenges associated, Precision Nanosystems have developed a suite of instruments to prepare LNPs at different scales using next-gen mixing technology. Next-gen mixing enables flow rates that are thousands of times higher than conventional microfluidic designs, while maintaining the controlled mixing conditions that are seen on bench-scale instruments. This circumvents the need to redevelop upstream processes at larger scales to yield a product of similar quality to that which was developed on the bench scale. To investigate the scale-up performance of the lead lipid mix composition used in this product, we formulated LNPs on different instruments using different next-gen mixers. We selected the instruments and mixers to fully encompass all scales that are required in the development of a pharmaceutical product. The specific nanoassembler instruments, mixers, and scales which are used in each test condition are shown in the table on the left, while all other critical process parameters, such as the flow rate ratio and entropy ratio, were maintained between test conditions and are shown in the table on the right-hand side. After mixing, all LNPs were subjected to the same downstream processes previously developed for large-scale batches. To summarize the downstream processing, buffer exchange and concentration of LNPs was accomplished by tangential flow filtration, and this was followed by sterile filtration. In this study, the same SARS-CoV-2 self-amplifying RNA used in previous datasets was encapsulated. And it should be noted that the color scheme used in the table on the left will be the legend for data shown in upcoming slides, where green-blue is used for bench scale instruments with darker shades indicating a scale increase, and purple being used for commercial scale instruments, where again a deeper shade indicates an increase in scale. It should be noted that the NextGen 500 mixer can be used on both bench and commercial scales, allowing larger batches to be prepared on commercial scales using the same mixer that was used in development. So after formulation, the LNP physicochemical characteristics were first analyzed as a function of both the mixer, or the cartridge, and the instrument itself. The graphs here show the size, PDI, and encapsulation efficiency from top to bottom. And as you can see, LNP size, polydispersity index, and encapsulation efficiency were maintained at all scales. The size varied from 60 to 86 nanometers, which are all within the expected range, while PDI measurements were consistently below 0.2, indicating that the LNPs were homogeneous. At all scales, the encapsulation was greater than 93%, indicating that even at large scales, waste of precious RNA will be minimized. Biological activity is another critical quality attribute of LNPs, and to determine the effect of scale on the in vitro expression of spike protein, we treated BHK 570 cells with decreasing doses of LNPs. The schematic on the left-hand side shows the transfection process, where the LNPs are first taken up by cells by endocytosis, and then the RNA is released into the cytoplasm by endosomal release, where LNP structure changes in response to pH changes inside the cell. The host cell machinery is then used to translate the mRNA into its protein, which is then transported to the cell surface for expression. After 24 hours, cells are stained with an anti-spike protein antibody, which binds to the protein on the cell surface. The antibody is conjugated with electrophoreate, and this fluorescence is detected by a fluorometer. The percentage of transfected cells was plotted as a function of self-amplifying RNA dose, as shown in the left graph. And from the best fit curve, the EC50, or the concentration required to transfect 50% of cells, was determined. Although small differences in EC50 were observed, specifically between the Ignite Plus and the commercial formulation system, no trends were observed that indicate a worsening performance with scale increase. So in addition to in vitro activity, it is also crucial to determine the in vivo activity of LNPs at all scales. To do this, the same immunization schedule used previously was used, but to briefly remind you, biopsy mice were injected intramuscularly with both a primary and a booster dose of LNPs on day 0 and day 28 at a dose of 1 microgram of self-amplifying RNA. Serum was collected on day 21 and day 42, and the humoral immune response investigated by quantifying the amount of spike protein-specific IgG present by ELISA. Robust immune responses were observed in all samples, and between each nanoassembler instrument, there were no significant differences at either day 21 or day 42, highlighting that the in vivo biological activity of the LNPs was preserved at large scale. These results, alongside the physicochemical characteristics and in vitro potency, indicate that the critical quality attributes of these LNPs were maintained across all scales when precision nanosystems instrument and mixer technologies were utilized. So that is it for the data that I'm presenting today. And to summarize, we wanted to showcase how this new product can accelerate your research in vaccine applications. Using our LNP kit and the associated protocols, you can confidently produce high-quality, stable LNPs with ease, streamlining both the screening and optimization of lead candidates through the preclinical stages of vaccine development. Additionally, we have showed that the LNPs produced using this product can be manufactured at the commercial scale using our nanoassembler suite of instruments, all while retaining the critical quality attributes of an LNP vaccine achieved at the bench scale. Before I finish, I would just like to take a moment and thank all of the contributors at Precision Nanosystems for their support and hard work throughout this project. It's very much appreciated. Okay, so moving on to the Q&A portion of the webinar. These are some of the questions that we commonly encounter. So the first question is, what challenges and hurdles are common to encounter in early preclinical stages of RNA-LNP development? And there's a few different challenges that I'm going to discuss. The first is the development of the active pharmaceutical ingredient or the nucleic acid. So you'll want to develop a couple of lead RNA structures that are potent and will effectively, they'll exert their required effect on the cells of interest. So there's different sequences that you can test, and they'll form different structures. So you really want to make sure that the RNA that you're developing is the best that it can be. And the LNP kit that we have developed and discussed in this presentation will help with lead candidate selection, as it can be used to screen all of the candidates that are under consideration. And the second challenge I'm going to talk about is LNP development. So the design of the lipid nanoparticle itself can have a huge impact on the efficacy of the payload. And the LNP composition itself needs to be optimized for specific applications. And this is where the ionizable lipid library that Precision Nanosystems have can be used, as different lipids can be selected based on their structures and characteristics, and can be used to fine-tune the potency of the lipid nanoparticles with your lead candidate RNA structures. As well as this, developing the process parameters of the formulation process is also very important. So different mixing ratios, different RNA to ionizable lipid ratios, or the NTP ratio. These things can have huge impacts on the properties of the lipid nanoparticle that you develop, and that can also be a challenge to develop, especially at the early stages. So moving on to the second question, how important is it to consider the properties of the ionizable lipids when designing lipid nanoparticles? So the short answer would be it is very important. And the longer answer, I'll talk about why it's important. So the ionizable lipid is arguably the most important component in LNPs, and it can have a huge effect on LNP potency. So really getting the structure of the ionizable lipid right for your specific RNA and application is of great importance. So the specific structure itself can affect both the biodegradability of the ionizable lipid and the LNP, and also the pKa is very important. So pKa is the pH at which 50% of the ionizable lipid is ionized. And this does need to be optimized for each specific application. So in general, the pKa should be between 6 to 7, although specific values can vary between different applications, and also for delivery to different cell types. So if the pKa is too low or too high, the efficient delivery will be impacted, either due to instability of the ionizable lipid or toxicity that occurs. And so moving on to the third question, what factors are important to consider when scaling up an LNP formulation? So the main problem with scale up is that you can have a brilliant LNP on bench scale, but if you cannot get that same potency at large scale, it can never be used, widely used to treat patients in clinical and commercial scales. So you really do need to look at the critical quality attributes of the LNPs and also the potency of the LNPs at various different scales to monitor how these are being affected by scale increases. So the mixing itself on large scale can impact the potency and critical quality attributes of the LNPs. But as you've seen in this presentation, we use the nanoassembler suite of instruments, and we have shown equivalent mixing at both small and large scales, resulting in similar CQAs and potency between bench and large scale. So if scale up is something that you're considering, definitely reach out to us because we have a lot of experience in making sure that the LNPs translate and are relevant at large scales. So as well as mixing, downstream processing is also important. So the downstream processing will be very different on the bench compared to on large scale, as it's much easier to do things on the bench when it's very small scale, but at large scale, you kind of need to automate things. So the processes usually need to change entirely, but we've taken a lot of the guesswork out of that and have de-risked it. And we know how to translate the small scale processes to large scale processes for this. And finally, it's all well and good if we're using research use only materials or at preclinical stage. If you cannot get these materials at GMP grade, you can't do anything with it. So all of the ionizable lipids offered by precision nanosystems are can be prepared at a GMP grade, and so we've taken that risk out of it as well. So moving on to the next question, what factors need to be considered when choosing a cryopreservation buffer? So the main point of the cryopreservation buffer is to effectively protect the LNPs at the desired concentration of injection for up to a few weeks. So for the preclinical stage, it's often quite stressful to prepare your LNPs for use within 1 or 2 days of preparation. So you need to get all of your analytics done in that time and make sure your animal studies are ready to go. So we wanted to take that stress out of the process, and we wanted to show that our LNPs can be stored for up to 1 week in the fridge. So it gives you a little bit more time to play with, and you can also store them at a -80 freezer for up to 6 weeks. So this allows you to allocate your LNPs for primary and booster doses. The same batch can be used. And also the LNPs can be formulated a few weeks in advance, making it a little less time sensitive. So that's why it's important to have a cryopreservation buffer, but in the development of a cryopreservation buffer itself, as I mentioned in the presentation, the specific type of buffer, the molarity, the cryoprotectant that's used, and the concentration of cryoprotectants all need to be optimized. And different LNPs have shown better protection when different buffers are used. So it is something that does need to be optimized. So really what we want to make sure is that we have a cryopreservation buffer that allows us to store the LNPs for up to 6 weeks to remove the time sensitivity of the formulation process. We also need to make sure that we can effectively protect the LNPs at the concentration at which we want to dose, because we don't want to then have to redilute or reconcentrate the LNPs prior to injection after we store. And osmolality is also an important consideration, as the mice will be injected with LNPs in this cryopreservation buffer. So we need to make sure that the animals are not going to come to any harm or pain as a result of this buffer. And in the development of this cryopreservation buffer, we have taken all of these factors into consideration. So finally, the last question is, could lyophilization be used to preserve these lipid nanoparticles? And absolutely, we do have experience lyophilizing LNPs at Precision Nanosystems. I didn't talk about it in this webinar as it's a completely different webinar in itself. So if you're interested in lyophilization, please do reach out to us for more information. But to talk a little bit about the differences between lyophilization and cryopreservation, absolutely, lyophilization is very advantageous to LNPs, and I believe it's where the future of lipid nanoparticles is going to move, as it does remove the requirement for cold chain. So large -80 freezers won't be needed for the next vaccine campaign. And they won't need to be transported to different areas using cold chain. So absolutely, transferring and storing lipid nanoparticles at room temperature is very advantageous to the field. In this product, we did not consider lyophilization, as to effectively lyophilize, you require a freeze dryer and knowledge of how to use this freeze dryer. So freeze dryers are not as commonly found in labs as a low temperature freezer. So the benefit to using a cryopreservation buffer as opposed to lyophilization would be that it is a much easier process to use, and you don't need to purchase any other expensive equipment and learn a whole new field in order to do so. So it's quick, it's easy, and it's relatively cheap compared to other options. So yeah, that's all of the questions that we have in this Q&A session. But if you have any more questions that we didn't cover, please feel free to reach out to us using the email address shown here. And for more information on our lipids, please follow the link shown in the slide here. And yeah, you can get some contact information there as well. So all that's left to say is thank you for taking the time out of your day to listen and learn more about our new products and technologies. And again, for any more information or any further questions, please reach out to us.